Ever wondered why phlebotomy tubes come in different colors? Well, it's not just to brighten up the lab. These colors play a crucial role in the world of medical diagnostics. Welcome to our journey into the realm of phlebotomy tubes. Phlebotomy, the act of drawing blood, is something that's practiced daily in healthcare settings worldwide. And those vibrant tubes, they each serve a unique purpose. You see, each colored top corresponds to a specific type of test or analysis. The color isn't just for show. It signifies the kind of additive present in the tube, which prevents the blood from clotting, or prepares it for a specific type of analysis. So whether it's a lavender top for a complete blood count, or a gold top for serum testing, each tube has a role to play. Intriguing, isn't it? Stay tuned as we delve deeper into the world of phlebotomy tubes and their fascinating color codes. You might be curious about the phlebotomy order of draw. And indeed, it's a fascinating process that requires precision and care. Let's delve into it. In the world of phlebotomy, the order of draw refers to the sequence in which blood collection tubes must be filled. This order is not random but is specifically designed to prevent cross-contamination of additives between tubes. Imagine you're a painter. You wouldn't dip your brush into multiple paint pots without cleaning it in between, right? The same principle applies to phlebotomy. Each tube in the order of draw has a specific purpose and contains different additives. These additives might include anticoagulants, clot activators, or other chemicals that prepare the blood for specific types of analysis. The phlebotomy order of draw typically begins with tubes for blood cultures, followed by coagulation tubes, and then tubes with various additives. These additives could be anything from EDTA, an anticoagulant that binds calcium ions, to heparin, which inhibits clotting, to sodium citrate, another anticoagulant that's commonly used for coagulation tests. But why is the order so important? Well, imagine you're drawing blood, and you first fill a tube containing EDTA, then you fill a tube for a coagulation test. The EDTA from the first tube could contaminate the needle and get into the second tube, affecting the coagulation test results. Following the correct order of draw is like following a recipe. If you miss a step or mix up the order, the final product might not turn out as expected. In the case of blood tests, this could mean inaccurate results, which could lead to incorrect diagnoses or treatments. And we certainly don't want that. The order of draw is a fundamental aspect of phlebotomy that ensures the accuracy and reliability of blood tests. It's a careful dance, a precise sequence that, when done correctly, helps healthcare professionals make informed decisions about patient care. Remember, following the correct order of draw is crucial in obtaining accurate and reliable results. And that's the essence of the phlebotomy order of draw. Now, let's explore the significance of tube additives in the phlebotomy process. Imagine you're making a cake. You have a recipe and each ingredient has a specific role. Flour gives the cake structure, eggs add moisture, and baking powder makes it rise. Similarly, in the phlebotomy process, we have our own recipe, and each tube additive has a unique function. Tube additives are substances added to phlebotomy tubes to either prevent blood from clotting or to preserve certain components of the blood, depending on the test that needs to be conducted. They are the unsung heroes of phlebotomy, ensuring that the blood sample remains in the desired state until testing. For instance, anticoagulants like heparin, EDTA, and sodium citrate are added to tubes to prevent blood clotting. These additives work by binding to calcium ions in the blood, an essential component for blood clotting. They ensure a free-flowing sample ideal for complete blood counts and blood films. On the other hand, tubes used for serum tests contain a clot activator. This additive promotes clotting, allowing the serum, the liquid part of blood after clotting, to be separated more easily. The serum is then used for various tests including hormone levels and drug monitoring. But here's the catch. Using the wrong additive can significantly affect test results. For example, using an anticoagulant for a serum test would prevent clotting, making it impossible to separate the serum. Similarly, using a clot activator for a complete blood count would cause the blood cells to clump together, leading to inaccurate results. So, much like our cake recipe, the phlebotomy process also requires precision. Each tube additive has been carefully chosen to create the perfect conditions for each test, and it's crucial for healthcare professionals to understand this to ensure they use the correct tube and additive for each sample. In the grand scheme of things, these small additives play a monumental role. They help maintain the integrity of the blood sample, ensuring accurate and reliable test results. So you see, each additive plays a vital role in ensuring the integrity of the test results. Each phlebotomy tube doesn't just go anywhere in the hospital or lab, there's a specific department for each one. The life of a phlebotomy tube is an interesting journey, as each one is destined for a particular department within a hospital or laboratory. These departments are specialized, with each one focusing on a specific type of sample analysis and testing. Let's start with the clinical chemistry department. This is where you'll find the serum separator tubes, or SSTs. These tubes typically contain a gel that separates the serum from the blood cells, and they're used for various tests such as liver and kidney function, electrolytes and hormones. Next, we have the hematology department. This is where lavender top tubes filled with EDTA as an anticoagulant are sent. They are used for tests like complete blood count, hemoglobin, and hematocrit. Then, there's the coagulation department. Here, light blue top tubes filled with sodium citrate are analyzed. The coagulation department is all about assessing the clotting ability of the blood, and these tubes are used for tests like prothrombin time and partial thromboplastin time. The microbiology department, on the other hand, deals with blood culture bottles. These are used to detect and identify microorganisms like bacteria and fungi in the blood, which are often the culprits behind septicemia and other infections. The immunology department receives the Tiger top tubes, which contain a serum separator and clot activator. These tubes are used for tests related to immune response, such as antibody detection and measurement. Lastly, let's look at the molecular diagnostics department. This department deals with tests at the gene level, focusing on identifying genetic and molecular alterations. They receive the yellow top tubes, which contain ACD solution, and are used for DNA testing. So you see, each department has a critical role to play in the analysis of phlebotomy tubes. From clinical chemistry to molecular diagnostics, every department contributes to the bigger picture of patient care and diagnosis. Every department plays a critical role in ensuring that each phlebotomy tube is analyzed accurately and efficiently. Let's wrap up by summarizing the key points about the phlebotomy order of draw. We've delved deep into the world of phlebotomy tubes today, discussing their significance, their additives, and the departments of analysis associated with each. It's a lot to take in, but these are the fundamentals that underpin the science and practice of phlebotomy. To start with, we talked about the color codes. Each phlebotomy tube has a specific color code, which signifies the type of test that it's used for. From gold top tubes for serology tests to lavender ones for hematology, these colors aren't just for show. They're a crucial part of ensuring that the right tests are conducted on the right samples. Next, we explored the phlebotomy order of draw. It's not a random sequence, but a carefully designed system that prevents cross-contamination between tubes. Remember, the order starts with blood culture bottles, followed by coagulation tubes, and then chemistry tubes. Each step has been meticulously planned to ensure the integrity of the samples. We also discussed the importance of tube additives. These substances, which range from anticoagulants to clot activators, play a vital role in preparing the blood samples for analysis. They can affect the test results, so they need to be chosen with care and precision. Lastly, we looked at the respective departments of analysis for each phlebotomy tube. Each tube type has a specific department where it's analyzed, from microbiology to biochemistry. This is another critical aspect of the process, as it ensures that each sample reaches the right place for the right test. In conclusion, every element of the phlebotomy order of draw, from the color codes and the sequence to the additives and the departments of analysis, has a crucial role to play. They're all interlinked, working together to ensure the accuracy and reliability of the tests. Remember, understanding these key points is crucial for anyone involved in the phlebotomy process. We've covered a lot of ground today, haven't we? From understanding the phlebotomy order of draw, to the significance of tube additives, and the respective departments of analysis for each phlebotomy tube, we've taken a deep dive into the fascinating world of phlebotomy. We hope you found this content informative and helpful. There's always more to learn, and a deeper understanding to be gained, and that's where we come in. We're committed to bringing you more insights and deeper knowledge, unraveling the complexities of the healthcare world, one video at a time. If you have any comments or questions about today's topic or any suggestions for future videos, we'd love to hear from you. So don't hesitate to leave a comment below. And if you want to keep up with our latest content, there's one simple thing you can do. Don't forget to hit that subscribe button and join us for more fascinating insights into the world of healthcare. Until next time, keep learning and stay curious.